Hello and welcome to the second session in a series of lectures on Chapter 10, Muscle Tissue. We last left off talking about some of the layers that surround muscles and muscle tissue. And I want to go ahead and just review that very, very quickly here. And then we're going to move into some new material. So as you recall, we had talked about layers of tissues, connective tissue that we use to wrap up muscles. We talked about the superficial fascia, the deep fascia, and then we moved into three layers in particular, which is the, as you can see here, the epimysium, the perimysium, and the endomysium. And I wanted to focus your attention on this area right here. So this represents a single muscle, biceps brachii, let's just say. It's connected through a tendon to the bone, and as we'll see, this muscle shortens given the right stimulus to flex the forearm. We're going to wrap this muscle, the entire muscle, with that first layer, this layer right here, the epimysium. And then within the muscle, which is composed of many individual cells, individual muscle cells. They're called muscle fibers. We're going to take some of those muscle fibers and bundle them up into groups. As you can see right here, there's a group here, there's one right there, here. So we're going to bundle the muscle cells into groups, and we're going to use connective tissue once again to do that, and that's going to be the perimysium. So that bundle of cells that you see right here is called a fascicle. That fascicle is literally composed of several different numbers of cells. So each fascicle can be a different size within the muscle. And the important point here is that we're wrapping individual cells. So we wrap the entire muscle with the epimysium, and then we're going to wrap bundles within that muscle, bundles of cells within that muscle. We'll call that a fascicle, and we'll use the perimysium. These two wrappings are dense, irregular connective tissue. When we look at individual cells, so we bundled up a bunch of cells, and then we're going to pull out one of those individual cells, which we have right here, and we're going to surround the individual cell with the last, the endomysium. This is areolar connective tissue. And here we're going to embed in this layer surrounding the cell, we're going to put our nerve, our motor neuron, and capillaries. So the capillaries are going to be embedded within that layer so that this cell can get what it needs and get rid of waste materials, and so that it can be stimulated by this motor neuron. These structures here are not cells. They're bundles of fibers inside the cell. Those bundles are called myofibrils. And this is where we're going, and this is one of the reasons I wanted to review this. We're going to start looking on the inside of the cell and taking apart these myofibrils to see how they're built and put together. But they look and can be mistaken for individual cells. No, this is the entire cell, and we're bundling these cells together into fascicles, and then we're wrapping the entire muscle. All right. Having accomplished that review, let's begin in our new material, and we're going to talk about now the neuromuscular junction. The neuromuscular junction. All right. So a couple of things. We're going to get some written notes down, and then we're going to do a lot of drawings to illustrate what we're dealing with here. So first, the notes. So a couple of points we're going to make. So first of all, we can say that this structure, the neuromuscular junction, is the point of contact between the nervous system and the skeletal muscle system. It's where the two come together. And what we need to understand is how that is put together, how that is constructed, and what's happening at that point of contact between the nervous system and the skeletal muscle system. So each individual cell has a single neuromuscular junction, usually located at the center of the cell. What do I mean by that? Well, if we draw a cell, remember that these cells are long cylindrical structures. The neuromuscular junction will be in the center, not here at the end, not here at this end, in the center. And why? Because by placing that structure in the center of the cell, the ends are equal distance from the initial point of stimulus. If I were to put it here, then this end of the cell would be very, very long from the initial stimulus that the neuron is going to provide. So we're centering the contact point in the center of that long cylindrical cell so that it is strategically located so that a signal that arises here is traveling to the ends of the cell, and those ends are equal distance from that point of contact. Okay? So, point of contact between the nervous system and the skeletal muscle system. That contact point is referred to as a synapse. Or as a synapsis. Okay? So you're going to hear this word synapsis multiple times. We're seeing it for the first time here, but when you talk about nervous tissue, and you've probably heard this, in fact, in your life when people talk about synapses in the brain, what they're talking about is a connection between two neurons. Now, this point of contact, to be clear, is there is no physical contact. No physical contact at the neuromuscular junction. So when we come in and look at that contact, there's a space that separates the neuron from the muscle cell. They're not physically touching each other. The distance there is extremely small, very small, but still a distance. Okay? So how does communication take place? How do we stimulate the muscle cell if there's no physical contact? What is the connection, the physical connection between this cell and that muscle cell? And the answer is a neurotransmitter. So the communication between a motor neuron and a muscle fiber is a neural is the what is called a neurotransmitter. It's a chemical, and typically what it is acetylcholine. Which we can abbreviate in the following manner. So that the physical communication between the neuron and the muscle cell is a chemical called a neurotransmitter, and it is acetylcholine for skeletal muscle cells. So let's talk about the motor neuron a little bit. The structure of a motor neuron. So when we look at a motor neuron, what we will see is, if we were to draw a very sort of sketchy picture of this, we would see a cell with a long axon. There would be some structures here, which we're not going to talk about. So roughly what we're seeing is something like this. And then we'll get this branching like this. So we get dendrites, which you'll learn about in the next section of this class, 40B. We're just going to name them here. We've got the cell body, and we have this very long axon. And then what we're going to have is an axon collateral, and then an axon terminal, and then a synaptic end bulb. So the order of movement. So what's going to happen, we're going to generate what is called an action potential here in the body cell. So we're going to stimulate this. And this cell body is typically in the spinal cord someplace. So the cell, the body of the cell, is in the spinal cord. But this process will leave the spinal cord and go out into the body and connect with the skeletal muscle somewhere in the body. So if we're looking at the foot or the hand, we have a cell body that's in the spinal cord, and this long process that we're calling an axon travels from the spinal cord all the way down the length of the arm and makes contact with muscles that are in either the forearm, in the hand, in the foot, wherever that muscle may be. And the order of divisions of this long process is axon, then there's an axon collateral, which then branches off into axon terminal, which ends in a synaptic end bulb. Okay. So if we were to write that down, it would be axon, axon collateral, axon terminal, synaptic end bulb. That's the order in which that signal, which originated in the cell body, is going to move. All right. Let's talk a little bit about the synaptic end bulb. So the synaptic end bulb. Ooh, there you go, synaptic end bulb. So if we were to draw this, here we go. So here's my axon terminal. And here's my synaptic end bulb. Inside the synaptic end bulb, we're going to find synaptic vesicles. These synaptic vesicles will contain acetylcholine. So let's go ahead and show us that. And let's bring in some color. So that's our acetylcholine. It is the release of acetylcholine which stimulates the skeletal muscle cell. It's going to be released in that space. So it's released into the space between the synaptic end bulb and the skeletal muscle cell. That space is referred to as the synaptic cleft. So that space is called the synaptic cleft. It contains a substance, so it's not just empty space. It contains a substance. It's filled with a gel-like substance. And that gel-like substance is referred to as the basal lamina. Okay. So, filled with a gel-like substance. And this is referred to as the basal lamina. So let's go ahead and show us that. We're going to fill that space. Which is the basal lamina. Let's focus on the muscle cell now in this space that we're calling the neuromuscular junction. So we've described basically the neuron and the important structures that we will need to know about. Let's focus our attention now on the muscle cell. and describe a little bit about what's happening there. So skeletal muscle cell structure at the neuromuscular junction. So the membrane, the plasma membrane of a muscle cell, is referred to as the sarcolemma. And at the junction of the synaptic end bulb and the sarcolemma, we get folding of the membrane. So that contact point at the, and I'm going to start abbreviating this, neuromuscular junction, is called the motor end plate. Okay. It is folded. And the foldings are referred to as junctional folds. And on the membrane, the sarcolemma, at the motor end plate, are proteins. Protein receptors. They bind to acetylcholine. So if I may, we're going to draw a picture now. Okay. So let's go ahead and we will highlight. I'm going to leave out the basal lamina for this, to make this less complicated. Okay. And we're going to bring in some proteins here. Okay. So now we can start labeling this whole thing. This is my motor end plate. These are my junctional folds. These things, the little blue dots that I drew in there, is my protein receptor. for acetylcholine. Okay. So next, we're going to talk about the steps that are involved in stimulating a cell. And before we do that, let's just review what we have been talking about here. So here is my axon terminal. There's my synaptic end bulb. There's my synaptic vesicles containing acetylcholine. Here is the synaptic cleft, the space, and that's filled with a substance. So there's my synaptic cleft filled with a substance. That's called the basal lamina. Here's my junctional folds. And this entire surface is the motor end plate. There's my motor end plate. It's the entire surface. And you can see that we're going to release the synaptic vesicles, or we're going to release the acetylcholine from the synaptic vesicles. It's going to diffuse across this space, going to bind to these receptors. These are actually ion channels for sodium. And so when we bind that receptor, or when we bind that receptor with acetylcholine, the channel opens and sodium will flow into the cell, and that's going to cause our action potential on the muscle cell. So we've got an action potential in the neuron that releases the synaptic, that releases the acetylcholine in the synaptic cleft, and the binding of that acetylcholine, that neurotransmitter, to these receptors, allowing sodium to move into the cell, will initiate a muscle action potential, or an action potential on the muscle cell. So we have two action potentials, one that initiates the release of acetylcholine, and a second one that's generated as a result of that on the muscle cell. Okay. So here is not the greatest picture in the world, but there's my synaptic end bulb, okay? And this is our muscle cell. So this is how it typically look at an actual neuromuscular junction. They're calling it a myoneural junction. I'm not sure where that terminology is coming from, but that's what you're looking at here. This is the neuromuscular junction. They're actually naming it properly there. Okay. All right, so let's go ahead and outline the steps that we've just talked about, and we'll put it in an order so that it makes sense to us. Okay, and then we'll have a drawing, an image that will allow us to relate it to the actual structures. Okay, so we're going to label this series of steps as the stimulation of a muscle cell by a motor neuron. Okay. Number one, we're going to refer to this step as the release of acetylcholine. Or ACH. Okay. And in this step, we will have nerve impulse, also referred to as an action potential, causes the synaptic vesicles to undergo exocytosis. This is going to release acetylcholine into the synaptic cleft. Acetylcholine then diffuses across the synaptic cleft and then we're into our next step. Step number 2 is activation of acetylcholine receptors. And this is going to involve binding of acetylcholine to receptors on muscle cell membrane at the motor end plate. The sodium ion channels, and these are the receptors, open. Sodium flows into the cell. This would be the muscle cell. Next step, number 3. Production of a muscle action potential. Okay. What we're going to get is the inflow, meaning as sodium flows into the cell, causes the inside of the cell to become more positive. The change in charge across the membrane triggers an action potential. In the muscle cell. the action potential spreads throughout the cell membrane. So it starts at the neuromuscular junction, but then from there it simply spreads out. It expands outward. Imagine throwing a stone into a pond, and where the stone hits the water, that would be your neuromuscular junction. And then you would notice a series of ripples that extended out from that single spot in a concentric set of circles away from that point of contact where the stone hit the water, and that those ripples would just expand outwards along the surface of the pond. The same thing happens here at the point of contact, the neuromuscular junction. As we change the charge from a negative inside the cell to a positive, that change in charge triggers this action potential, which begins there at the neuromuscular junction and then spreads out like those ripples in a pond along the surface of the cell membrane. The action potential will trigger, let's put it that way, the release of calcium from the sarcoplasmic reticulum of the muscle cell. The release of calcium causes a muscle contraction. So starting at the neuron, we release acetylcholine. Acetylcholine binds to to the receptors. The receptors open. Sodium flows into the cell. That flow of positive charges into the cell changes the charge across the membrane in such a way that it triggers an action potential, which then spreads out along the surface of the membrane, down the T-tubules. That action potential is communicated to the sarcoplasmic reticulum, and we release calcium, thus causing a muscle contraction. Okay. Step number 4. Termination of acetylcholine activity. Okay. So, the motor neuron action potential stops. In other words, we no longer have an action potential being generated in the motor neuron. That is going to stop the release of new acetylcholine into the synaptic cleft. Okay. Acetylcholine is broken down by enzyme activity. So the acetylcholine that's already there is removed because it's broken down by an enzyme. And wait for it, this is the name of the enzyme. Acetylcholine esterase. So acetylcholine esterase. And that's going to end stimulation of the muscle cell. That will end the action potential on the muscle cell membrane. Calcium is, let's put it this way, the release of calcium from the sarcoplasmic reticulum stops. Calcium is taken up into the sarcoplasmic reticulum. It's pumped back into the sarcoplasmic reticulum, and muscle contraction stops. Okay. So let's take a look at this whole set of things we've just talked about using an image. And this is one of the images that you should have downloaded from Canvas. And I'm going to go ahead and bring that image up so we can see it. And we'll walk our way through this process that we just talked about. Okay. So starting right up here, we're going to get an action potential. From the motor neuron. That's going to cause the release of acetylcholine. We're going to bind. Acetylcholine binds to receptors. And I'm going to use pink. That's going to be our sodium. Sodium flows into the cell. So sodium flows into the cell. That's going to change the membrane potential, the difference in charge across this membrane, because we're bringing in sodium, which has a positive charge. So let me step back a moment. We'll come back to this drawing, but let me step back a moment and explain a little bit about what's happening here. I'm not going to go through the entire lecture on an action potential, but we need a little bit more information here to understand this process. So let me go there and show you what's happening here. So what's happening here is that there is a difference in charge. So typically the inside of the cell is negative and the outside is positive. So this is outside or extracellular, and this is intracellular. And what we're going to do is we're going to bind, and I believe I was using this color, we're going to bind to these receptors, acetylcholine. And that's going to allow sodium to flow into the cell. So initially positive on the outside, negative on the inside, but now we're adding these positive charges, so that's going to reverse. So it will no longer be negative compared to the outside. It will be positive compared to the outside. That change in polarity is going to trigger an action potential. So the change in charges is going to trigger an action potential, which will then spread out from this contact point in all directions. Okay. Now, I have not explained what an action potential is, and I don't intend to at this point. You need a lot more background and information before that would make sense to you. You will get that in 40B. So right now, we're just treating this action potential as some sort of stimulus that is caused by the change in polarity across the membrane of the cell by allowing sodium to flow into the cell, triggering the so-called action potential. With that in mind, notice that it requires the membrane for that action potential. We cannot have an action potential unless we have a membrane. And again, keep it in mind, and let's take a look at our model. That cell is a big fat thing. And the action potential that we're talking about is happening, let me do it this way, just on the surface of the membrane. We need to get it inside the cell. The entire cell needs to be stimulated. But right now, all we've got is a message that is sitting on the surface of the cell. How do we get that message into the center of the cell? By the T tubules, because that is actually a membrane, isn't it? So that message on the membrane will travel down because this is in fact a membrane. And notice that that membrane is located directly next to the sarcoplasmic reticulum, which contains the calcium. So I'm getting that message into the center of the cell by these T tubules, and then the information can be transferred to the sarcoplasmic reticulum, and calcium can be released, causing a muscle contraction. Okay. So now, coming back to our image, we're going to generate an action potential. that action potential will travel down the T-tubule and be transferred to the sarcoplasmic reticulum. So the action potential is moving down the T-tubule, and the sarcoplasmic reticulum will release calcium. And so we get the calcium, and I haven't picked a color for calcium, I don't think, yet, so I'll do that now. Let's just use green. So now we've got calcium flooding into the cell from the sarcoplasmic reticulum. That's going to cause a contraction. Now, to stop the muscle from contracting, we're going to stop the action potential. We're going to stop releasing acetylcholine. The acetylcholine, which is present already, will be broken down and removed. The receptors will close. No more sodium flows in. We come back to the normal condition of charges across the membrane. That stops the action potential. That stops the release of calcium. With no calcium, there is no muscle contraction. Okay? All of that is happening in a split second. And at the end of all of this, as we talk about the next step, which will be actually what calcium is doing and how we generate that contraction, we'll understand that process, why calcium is so important in this process. And then I will summarize the entire thing again, and I'll emphasize to you how all of these steps, which have taken now, just this one part has taken about an hour's worth of explaining to you. And then this will be at least another hour to explain this process that's happening here after we release calcium. But all of that is going to happen just like that, just that fast. So it's a pretty amazing process, and you really should be pretty impressed with your body, because all of these steps are happening so quickly that you are capable of grabbing a set of keys. You ever had a set of keys or something, object on the table, and you see it's falling off, and boom, you reach down and you grab it before it hits the ground? Well, all of this has happened to allow that to take place, and much more on top of that, which I'll talk about when we're finally completed all of our understanding of a muscle contraction. So it turns out this takes longer than I thought it was, so we're going to stop right here. And what we're going to do next in the next session is we're going to talk about the structure inside the cell. We're going to talk about the sarcomere. We're going to talk about the proteins that make up the sarcomere, how they're arranged, and what they're for. And then we'll go through step by step and look at what happens when we actually shorten the muscle, when we have a muscle contraction. So ladies and gentlemen, it's been real, and I will see you in the next session.